So in this video, we are going to discuss RNA. Now we've talked about DNA and how DNA is a cookbook, but just like cookbooks contain hundreds of recipes, your DNA contains thousands of recipes. And remember, each recipe is a gene, and genes are recipes for polypeptides, which are the same thing as proteins. So when you talk about you have a gene for something, that means you have a recipe that tells you how to make a certain protein. But just like when you go to cook something, you don't need every single recipe in that cookbook. Your cells don't need every single gene on that piece of DNA. So when we talk about genes being expressed, those are the recipes that that cell happens to use. So brain cells will express different genes than stomach cells will express. So these are the recipes that they use. So whenever the cell needs to make a protein, it does not need the entire cookbook. And so when we talked about DNA, we talked about nucleotides and the number 3. There's 3 parts to a nucleotide. You have phosphate, sugar, which is deoxyribose, and a nitrogen base. And so like Legos, they stack on top of each other, and the sugar phosphates make up the sides of the double helix. So those are like the book covers. And so just like book covers protect the pages on the inside, and the pages are the important parts that you read and you learn from, the sugars and phosphate protect the genes, and the genes are your nitrogen bases. And remember, every 3 nitrogen bases on a piece of DNA is called a triplet. And so today we're going to continue on with this number 3. We're going to talk about RNA. There's 3 types of RNA. There's going to be 3 steps to making RNA. There's going to be every 3 letters again. And there's going to be 3 differences between DNA and RNA. So just as a review, if you look back at your DNA notes, when we make DNA, it's called DNA replication. And the only time you should do DNA replication is when you are doing cell division, either mitosis or meiosis. And in DNA replication, you are cloning, you are photocopying every single recipe in your DNA. Because in the end, you want to have 2 exact copies of DNA. And so again, we talked about how there's 3 steps. We unwind our DNA, so it's like you opening up your cookbook, and the enzyme that unwinds DNA is DNA helicase. Then you're going to photocopy, you're going to copy each recipe, and the enzyme that does that is DNA polymerase. I forgot for a second there. And then you have to seal everything back up and glue everything together, and the enzyme that does that is DNA ligase. But whenever your body needs to make a protein, like I said, when you go to make something, you don't use every recipe in that cookbook. And remember, our DNA is locked up tight. It's in this nice safety deposit box that we call the nucleus. So whenever your body needs to make a protein, you've got to go to the bank, you've got to unlock your safety deposit box, and then you've got to find the recipe you need. You do not need to carry the cookbook, the entire cookbook with 80,000 plus pieces of paper in it, to your kitchen. You just want one recipe. And remember, your DNA is very fragile. It's written on that papyrus paper. It's been in your family for generations, and you don't want to risk it getting messed up. So in your body, when you want to make a protein, you go to the bank, you find that special recipe, and you take a picture of it. And then you bring the picture back to your kitchen, and you use the picture in order to follow the instructions. That way the cookbook stays nice and safe and locked away so it doesn't get damaged. Well, this is where RNA comes into play. DNA is your cookbook, and DNA is the cookbook that our body uses to make proteins. And so anytime your body needs to make a protein, it will go to the DNA, and just like a book, it will open it up and find the recipe that it needs, and it makes a photocopy of that recipe. So instead of carrying the entire cookbook back to your kitchen, you just have one sheet of paper that you've printed off that has the recipe on it. And remember, the kitchen that we're going to be using are the ribosomes, because that's going to be the part that makes your proteins. But again, we're going to remember the number 3. So like DNA, RNA is a nucleotide made up, or excuse me, is a nucleic acids made up of nucleotides. However, again, the number 3. RNA has 3 differences from DNA. Now, as you can see in this picture, you can already tell that their shapes are going to be different. Because remember, structure determines function. RNA is different than DNA, so it's going to have a different shape. Now, so speaking of that, DNA, as you know, is double-stranded. It's like a book with 2 covers. RNA is one single strand. It's basically, I've gone onto the computer. There's millions of recipes for chocolate chip cookies I can find online. I find the one I want and I print it out. Well, that printed out copy is a piece of RNA. Also, DNA gets its name because it's made from deoxyribose sugar. RNA gets its name because it's made from ribose sugar. And you know that this is a sugar because it ends in ose. And you can look at this picture and you can see deoxy means it's missing an oxygen, just so you'll know. Well, this is what makes deoxyribose different from ribose. And then difference number 3 is the nitrogen base. In the case of DNA, you know that we use A's, T's, C's, and G's. In RNA, we use A, U's, C's, and G's. Okay. So C and G still goes together, but this time A and U go together. And what scientists believe is that, remember, DNA should not leave your nucleus. RNA has to leave your nucleus because RNA has to be able to go to the kitchen, which is the ribosome, so that the proteins can get made. So it's believed that nature needed a really quick way to look at a piece of nucleic acid and say, hey, are you DNA or RNA? Are you where you're supposed to be? So by having different sugars, and ribose is more stable than deoxyribose, by having a different sugar and a different nitrogen base, scientists believe it's just a quick way that our bodies could say, hey, are you where you're supposed to be? So again, we got number 3. 3 parts to a nucleotide, a sugar, a phosphate, and a nitrogen base. We've got 3 steps in making DNA, which is called DNA replication, which only happens during cell division. And we've got 3 differences between DNA and RNA. RNA is overall the copy of the instructions of DNA. Now you actually have 3 types of RNA. And so a lot of times if you look at notes about RNA, it's just talking about one type in particular. But we're going to look at all 3 types of RNA in this video. But if you think, like I mentioned, DNA is this cookbook with thousands of recipes in it. And you only need to print off 1 recipe. Well, that's going to be 1 kind of RNA. And so then instead of taking that entire really fragile, really special cookbook to your kitchen and then having to turn around and drive it all the way back, you can just print out the copy that you need and use the copy. And if the copy gets messed up, big deal. You just print out a new copy. But remember, if your DNA gets messed up, which is what a mutation is. Remember, mutations are when we change our DNA in some way. So we change the recipe. So instead of having sugar in the recipe now says salt. The problem is, remember, your body doesn't know that a mutation took place. So it follows the recipe like it's supposed to, but it makes the wrong protein. So not only is it wasting energy making something it doesn't need, it doesn't make something that it needs. And so that can cause that cell to die or be damaged in some way. But when you need to make a protein, then you're just going to go and you're going to print out, you're going to photocopy, you're going to take a picture of the 1 recipe you need. And you're going to see that that photocopy is just one of the 3 types of RNA that we're going to deal with. And then you take that copy to your kitchen, which is the ribosome, and you follow the instructions on that piece of paper in order to make your proteins. So, like I was mentioning, there are 3 kinds of RNA. And the first kind of RNA is what's called messenger RNA, and you'll hear me abbreviate that as mRNA. With messenger RNA, that's the photocopy you make. It's carrying the message. Okay. So whenever your body needs to make a protein, it goes to the nucleus and it opens up the DNA using an enzyme, which we're going to talk about. It's going to open up the DNA and then photocopy just the 1 recipe it needs. And then it closes the cookbook, locks it away so it's protected, and then you take that piece of paper with you to the kitchen. Well, messenger RNA is the photocopy or the picture or the printed off copy of the recipe. So it's carrying the message from the DNA to the ribosome saying, hey, this is the protein I need you to make. Messenger RNA is only used 1 time because it does, once you're done with it, you don't want it floating around. You're not going to reuse it because you might not need to make this protein all the time. And so you don't want to leave that recipe just kind of floating around because it can get mutated. Just like if you photocopy something, you just don't leave it sitting around on your table. You don't know when you're going to use it again. It could get torn, it could get stained. And with the technology we have today, you just take a new picture or you just photocopy it or you print it off again. So whenever messenger RNA is finished, your body does destroy it. And again, that's to make sure that it doesn't get mutated before you use it again. So most of the time when we talk about RNA, this is the type of RNA that the textbooks or the videos or professors are talking about, because this is the most common type of RNA that we make. Every time you need to make a protein, you will make a copy of messenger RNA. The other two types of RNA are reusable. You don't have to make them as much. And when we talk about making RNA, all RNA is made the same way. They're the same 3 steps. Enzymes tell your DNA what to do. And that's going to be a process that we'll talk about later that's called transcription. The second type of RNA that we have is called transfer RNA or tRNA. I call these guys our measuring cups. Whenever you need to get ready to cook something, you want to make sure you have all of your ingredients. So you get everything measured, you get everything laid out, you get what you need. Well, in the case of transfer RNA, their job is to gather the ingredients and transfer them. And remember, when we make proteins, our ingredients are amino acids. So you have 20 different types of amino acids, and with those 20 different types of amino acids, you have actually 64 different types of transfer RNA, which we're going to talk about again in a moment. With transfer RNA, you can see this one is reused, just like you'll use a measuring cup. You don't throw it away when you're done with it. You can reuse it over and over and over and over and over again. And transfer RNA is the same way. Now, eventually your measuring cup will get cracked or it gets so old that the numbers wear off, and you throw it away and you buy a new one. Your cells are the same way. After a while, transfer RNA just kind of gets old, it kind of gets frayed, so your body will break it down and will make a new one. But messenger RNA is the photocopy of the recipe that I'm going to read to make the proteins. Transfer RNA's job is to carry amino acids from the cell, where they are made from digesting food or other chemical processes, to the ribosome. So just like when you get ready to cook, you want to have all of your ingredients laid out in front of you. When your cell gets ready to make a protein, all these transfer RNAs just kind of line up, and they're just standing there with their ingredients, so they're ready in case you want to make them. Now this is where another group of 3 comes into play. When we talked about DNA, we talked about these things called triplets. And triplets were 3 letters on a piece of DNA. So like AAA, TTT. And remember, every 3 letters equals a word. So just like when you were learning to read, you just all of a sudden you recognize the words, or you recognize the letters, and then those letters turn into words. Remember, genes are sentences that your body reads to make a protein. And you can divide them into groups of 3s, and each group of 3 tells you an ingredient that you need. And so when it's on DNA, we call those groups of 3 triplets. Well, we can't call them triplets when they're on RNA because they're made differently. By definition, a triplet has a nitrogen base, a phosphorus, and deoxyribose sugar. Well, RNA is made from ribose sugar. Remember, structure determines function. So there's still 3 groups of letters. We just have to name them a little bit differently. So when you have 3 groups, 3 letters on a piece of DNA that tells you what ingredient you need, we call that a triplet. Well, 3 letters on a piece of messenger RNA that tells you what ingredient you need is called a codon, C-O-D-O-N. And 3 letters on a piece of transfer RNA that correspond with that ingredient is called an anticodon. And so remember, they all match each other. So if my DNA says ATG, my fault, the piece of messenger RNA that matches that will say AUC, and the piece of transfer RNA that matches that will say AUG. They all mean the same thing, but I have to call them 3 different things because they're found in 3 different places. So again, 3 differences between DNA and RNA. There's 3 kinds of RNA. There's 3 letters on DNA that mean something. There's 3 letters on messenger RNA that mean something. And there's 3 letters on transfer RNA that mean something. The third kind is called ribosomal RNA. And this is one that's not talked a lot about, especially in biology classes, because we don't make it. If you think about it, your DNA has 2 sides to it, a left side and a right side. And when we make DNA, we photocopy recipes, or when we make RNA, we photocopy recipes from DNA. Well, messenger RNA is made using one side of your DNA, and transfer RNA is made using the other side of your RNA. Well, the third kind of RNA, where does it come from? It's made by the nucleus. And if you remember, the nucleolus is an ancient bacterial-like organelle. It has its own DNA. And for years, scientists didn't know what the nucleolus did. So when they got the technology to take the nucleolus out, the cell lived for a little while, and then the cell started to die. So they started to look to see what was going on with that, and they realized that after a while, just like buildings fall apart and we bulldoze those buildings and we rebuild them, after a while the ribosomes in your body just biodegrade. And cells that did not have the nucleolus never replaced the ribosomes. And without the ribosomes, it couldn't make proteins, so the cells died. So it immediately let us know that the nucleolus had something to do with making ribosomes. And the nucleolus has its own DNA, and so it makes its own RNA. And its RNA is called ribosomal RNA. And ribosomal RNA are the instructions to basically tell us how to build the kitchen in the first place. So your DNA makes messenger RNA every time you need to make a protein. It will photocopy a recipe. That's messenger RNA. Your DNA makes transfer RNA, but transfer RNA doesn't have to be made as often because it can be reused numerous times. The nucleolus DNA makes ribosomal RNA. They're all made the exact same way. So when we talk about this process called transcription, same 3 steps, it's just depending on which one am I making at the time. So there will be some different enzymes involved. So all of this leads to what we call protein synthesis. And again, we've been talking about the number 3. Okay. Well, protein synthesis means we're making proteins. And so we're going from DNA to making a protein. But remember, proteins are made at the ribosome, and DNA is stuck in the nucleus. So DNA cannot directly make proteins. So you're going to see that there's actually going to be 2 steps. The first one is called transcription. And transcription is when your body makes makes a piece of RNA. Now we're going to use messenger RNA as our main example, but all RNA is made pretty much the same way. And how our bodies make RNA is in a process called transcription. And you will see it has 3 steps, and it has almost the exact same 3 steps as DNA replication. In DNA replication, we unwound our DNA, we then copied the recipes, and then we wound our DNA back up. Same thing is going to happen here. We're going to unwind our DNA, but we're only going to unwind 1 page. We're going to photocopy our DNA, but instead of photocopying every single recipe, we're only going to copy the 1 recipe we need. And then we're going to close our DNA back up, wind it back up, and we're going to make sure that our page is the way that it's supposed to be. So we're still going to have 3 steps, but we're going to have to use different enzymes. Because your DNA has to know, okay, am I photocopying every single recipe and making a new piece of DNA, or am I only photocopying 1 recipe and making some kind of RNA? Then we have what's called translation. Oh, and transcription happens in the nucleus because you're using DNA and making photocopies of it. Translation is where you actually make the protein, and this is what happens at the ribosome. And again, you will see that there are 3 steps to translation. We will call them initiation, elongation, and termination. But just remember the number 3. There's 3 letters in DNA, there's 3 letters in RNA. There's 3 steps to making DNA, there's 3 parts of a nucleotide, there's 3 differences between DNA and RNA, there's 3 kinds of RNA, every 3 letters mean something, 3 enzymes are used in each of the processes, there'll be 3 steps to transcription, and there'll be 3 steps to translation.